I want you to stop what you're doing for a second. If you're driving, keep your hands on the wheel. But if you're safe to do so, look at your thumb.
Okay. Looking at my thumb. I'm intrigued. Where is this going?
I want you to try to bend just the tip of your thumb back. Can you bend it back on its own, like almost 90 degrees?
The classic hitchhiker's thumb.
Exactly. Now, while you're contemplating your thumb's flexibility, think about your earlobes. Are they attached directly to the side of your head, or do they, dangle free?
You are taking us right back to ninth grade biology class, aren't you? The Punnett Square worksheet with the little pictures.
I am. But here's the problem with how we usually remember that class. We tend to think about these things in isolation.
Right.
You have the thumb gene, you have the earlobe gene, you have the can you roll your tongue gene. We treat them like separate little apps on a phone.
The monohybrid view. One trait at a time. It's simple, it's clean, it's easy to teach.
It's easy to teach, but nature is not simple or clean. When I look in the mirror, I don't see things one trait at a time. I'm not just a walking thumb.
I should hope not.
I am a thumb and an earlobe and a specific eye color and a certain height and, weirdly, a predisposition to sneeze whenever I look at the sun.
The photic sneeze reflex, that's a real thing.
See? I am a package deal. A messy, complicated package deal.
You're a messy, complex combination of literally thousands of traits all happening at the same time. And so the real question isn't just, how do you get the thumb? The much more interesting question is, how do you get the thumb and D, the free-hanging earlobes, and D, the blue eyes, all in the same person?
That is the mission for today's deep dive. We are stepping up from the minor leagues. We are done with the simple tall versus short conversation. We are looking at the dihybrid cross.
This is where it gets good.
And I have to be honest, when I first started opening the source material for this, the lecture notes, the diagrams, the probability formulas, I got a little intimidated. My high school biology trauma started kicking in.
Oh, I get it. You
Yeah.
see these giant grids, these 16-box squares or 64-box squares. It starts to look more like an algebra exam than biology.
It really does. But the promise you made me before we hit record is that there is a logic to the chaos. A beautiful logic, you said.
There is.
And that we can actually predict the future of a genome without needing a PhD in statistics.
We absolutely can. And, the math we're going to talk about today, it's not just an academic exercise.
Yeah.
It is the fundamental reason we understand how diversity works. It explains why you might have your mother's eyes, but your father's chin. It explains how nature shuffles the deck.
Shuffles the deck is a phrase that appeared a few times in the notes we looked at. I really want to unpack that. But before we get to the shuffling, I guess we need to look at the cards Mendel was playing with.
Let's look at the cards.
So the sources take us, as they always do, back to Gregor Mendel, the father of genetics. And he wasn't looking at thumbs or earlobes. He was looking at peas.
Good old-fashioned garden peas.
I always wonder, why peas? Why not something cooler, like racehorses or something?
Well, a few reasons. First, peas grow really fast. You get multiple generations in a single year, whereas with racehorses,
Right.
you'd be waiting a while.
Right. That makes sense.
They also have tons of offspring, so you get
Yeah.
a large sample size.
Okay.
But most importantly, and this was key for Mendel, they have very clear binary traits.
Binary meaning one or the other, no in between.
Exactly. The flower is either purple or it's white. The stem is either tall or it's short. There's no kind of medium height or sort of lavender-ish with Mendel's peas. It simplifies the variables dramatically.
Okay, so it was a smart choice. And up until this point in our reading, Mendel had been lucky. He picked these simple traits, but then he decided to make his life more difficult. He decided to track two things at once.
He leveled up. He went from a monohybrid cross to a dihybrid cross.
Let's get our definition straight before we really get into weeds here.
Yeah.
Dihybrid. What are we actually saying when we use that word? Because hybrid creates a specific image in my mind, and it usually involves a Prius.
Slight lap, a little different, yeah. So let's break it down. Di just means two. And hybrid, in a genetic sense, is just another word for a heterozygote.
Okay, pause on heterozygote. I always mix this up with homozygote. Can we just nail this down once and for all?
Absolutely. Think of the roots. Homo means same, hetero means different.
Same, different, got it.
In genetics, you get two copies of every gene, one from your mom, one from your dad. These copies are called alleles. If you get two of the same allele, like two tall alleles, you are homozygous for that gene.
You're a true breeder, as the texts call it.
Exactly. Your kids will only get tall alleles from you. But if you have one tall allele and one short allele, you have two different alleles.
So I'm heterozygous.
You're heterozygous. You are a hybrid for that one trait.
So a dihybrid is just someone who is heterozygous for two different traits at the same time.
That's it. You aren't just carrying a hidden gene for height, you're also carrying a hidden gene for, let's say, flower color. You are a double agent, a double carrier.
Okay, that clicks. So Mendel looks at his peas, and he picks two very specific traits to track for this big experiment. And for everyone listening, you should really visualize this, because we are going to be living with these two traits for the next hour or so.
Right. Trait number 1 was seed shape.
Which comes in two distinct varieties.
Whether it's perfectly round and smooth, or it's wrinkled.
And the sources are very clear that round is the dominant trait here.
It is. So we use a capital R to represent the allele for round, and a little r for the recessive wrinkled allele. If you have even one of those capital R's, your seeds are going to look round.
Even if you're secretly carrying the wrinkled gene.
Exactly.
Yeah.
Dominance masks the recessive.
Okay, that's trait 1. Shape. Trait 2 is seed color.
Which was either yellow or green. And this is shocking to some people because we all think of peas as being green, but yellow is actually the dominant trait.
Really?
Yep. So we use a capital Y for yellow and a lowercase y for green.
So if I'm a dihybrid for these two specific traits, my phenotype, what I actually look like, is round and yellow.
You look round and yellow because those are the dominant traits. But your genotype, your genetic code, tells a much more interesting story.
My genotype would be big R, big Y, little y.
Precisely. You are carrying the genetic potential to make wrinkled and green seeds, and it's just hiding there in your DNA, waiting for the right moment.
Okay. I think I've got the setup. We have a plant that is holding on to these secret recessive traits. Now, the sources mention this concept of the two monohybrids, and this felt like a bit of a cheat code to me when I was reading it.
It is the ultimate cheat code. It's the insight that makes all the complicated math simple.
And the idea is that even though we are looking at two traits at once, they don't know about each other. Is that the right way to think about it?
That's a perfect way to think about it. We tend to assume, oh, this is a single plant, a single organism, so its shape gene must be talking to its color gene.
Right, like they coordinate.
But Mendel's great realization was that they are operating in parallel universes. The gene that determines if the seed is round couldn't care less about the gene that determines if it's yellow. They are inherited independently.
So it's not like juggling two balls at once, which is hard. It's like juggling one ball and then separately juggling another ball.
And that means, mathematically, we can treat a dihybrid cross as two separate simple problems and just combine the answers at the end.
That seems way too easy.
It is. But, and this is a big but, to really believe that, to really trust the math, we have to see the experiment play out the hard way first. We had to see what Mendel saw.
The hard way. You mean the big, scary 16-box square?
I mean, the full Mendelian nightmare. We have to get our hands dirty and breed these things.
All right, let's go to Mendel's garden. We have to start with the parents, the P1 generation.
And in true Mendelian fashion, we start pure. We start with true breeders. These are plants that are homozygous for the traits we're interested in. So parent 1 is homozygous dominant for both traits.
So genetically, this plant is big R, big R, big Y, big Y.
Correct. And phenotypically, it makes only round and yellow seeds.
The dominant champion. And Mendel crosses it with its total opposite, parent 2.
Parent 2 is homozygous recessive for both traits. So its genotype is
Little r, and little y, little y.
And it looks.
It's a wrinkled green seed, the recessive underdog. Okay, so we have Mr. Dominant and Mr. Recessive. They cross. Pollen goes from one flower to the other. Seeds are formed. This creates the children, the F1 generation.
So what do they look like? What's their genotype?
Well, let's trace the DNA. Parent 1, the RYY parent, can only make one kind of gamete. It can only donate a big R and a big Y. That's all it has to give.
Right.
And parent 2, the RO parent, can only give a little R and a little Y. So every single offspring, without exception, receives one of each. Every single kid is big R, big Y, little Y.
RYY. They're all perfect dihybrids.
And since round and yellow are dominant, every single one of these F1 plants looks exactly like the dominant parent. They are all round and yellow.
100%. If you were to walk into Mendel's garden at this point in the experiment, you'd be pretty disappointed.
Yeah, you'd look at the crop of seeds and say, hey, Grigor, where did the wrinkled green ones go? Did we lose them? Did your experiment fail?
It looks like the recessive traits were just completely erased from existence.
Yeah.
Wiped out.
But we know they are there. They're just hiding.
Right.
And this is where Mendel does the thing that defines his genius. He doesn't stop. He takes these F1 plants, these identical-looking yellow round seeds, and he crosses them with each other.
Well, even simpler. He lets them self-fertilize.
Selfing. Which still sounds a little narcissistic for a plant.
Slight laugh. It's incredibly efficient. He just takes the pollen from an F1 flower and puts it on the stigma of the very same F1 flower. He's crossing RRYY with another RRYY.
Okay, this is where the chaos begins. The notes we have call this the gamete explosion. And I want to slow way down here because this is where I think a lot of people get lost in biology class. It's easy to write down the letters, but what is physically happening inside that plant?
Okay, let's personify it. Think about it from the perspective of that RRY plant. It needs to make a gamete, a sperm cell in the pollen, or an egg cell in the ovule.
Right.
It has to pack a genetic suitcase for its potential child. And the rules are, you must pack one shape gene and you must pack one color gene. You can't leave those slots empty.
Okay, I'm the plant. I'm packing my suitcase. I look at my shape shelf. I have two options, a big R allele and a little R allele because I'm a hybrid.
You have to pick one. It's a 50-50 choice. Which one goes in?
I'll pick the big R.
Okay, the big R allele goes in the suitcase. Now, move to the next shelf, the color shelf. You have a big Y and a little y.
Now here's the million-dollar question that defines this entire episode. Do I have to pick the big Y just because I picked the big R? Like, do the dominant genes hang out together? Are they buddies?
No, that is the whole point of independent assortment. The color shelf has no idea what you just took from the shape shelf. They are completely independent decisions. You can pick the little y.
So I could pack a suitcase with a big R and a little y.
That's one possible gamete you can make. What are the others?
Well, I could have picked big R and big Y. That's another. Or I could have started with the little R from the shape shelf.
Right.
And paired it with big Y. Or paired it with little y.
So how many total combinations is that?
That's four. 4 possible suitcases I can pack.
4 distinct combinations of alleles. The gametes can be RY, 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 or RY.
And since I'm breeding with myself, the mother side of the flower is making those same 4 types of gametes, and the father side of the flower is making those same 4 types of gametes.
Exactly. So now you have 4 types of pollen, each with a different genetic makeup, floating around and trying to fertilize 4 types of eggs, each with a different genetic makeup.
And this is why the Punnett square gets so big. In the simple monohybrid version, it was just 2 gamete types times 2 gamete types. 2 by 2 grid, 4 boxes, easy.
But now it's 4 gamete types by 4 gamete types.
A 16-box grid.
16 potential outcomes, 16 potential genetic futures for that little pea seed.
I have such a vivid memory of having to draw this in school. You make the big 4x4 grid. You write RY, 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 RY along the top. You write the same 4 down the side, and then you grind it out.
It's tedious work.
It is. You fill in the first box, big R from the top, big R from the side, big Y from the top, big Y from the side, R, R, Y, Y. Then the next one, R, R, Y, Y. It feels like accounting, not biology.
It does. But if you actually do it, and I encourage listeners to visualize this or even just doodle it on a napkin right now, a really beautiful pattern starts to emerge from all that ink. You start to see clusters.
You see the famous ratio, the one that, if you remember anything from this topic, you remember this, the 9.3.3.1 ratio.
The phone number of Mendelian genetics.
Let's decode that phone number.
Yeah.
What does the 9 stand for?
The 9 represents the most common outcome. If you go through and count up all the boxes, you will find that 9 out of the 16 squares will have at least one capital R and at least one capital Y.
So phenotypically, they look round and yellow. They look just like their parents.
Right. Most of the kids look like the dominant F1 parents. That makes intuitive sense. 9 out of 16, that's a little over half.
Okay, so that's the 9. Then we get to the 2 3s.
These are the interesting ones. These are the recombinants. This is where you see the deck being shuffled.
Recombinants, meaning new combinations that we didn't see in the original P1 parent.
Exactly. You will find 3 boxes where the resulting seed has at least one big R, but is homozygous for little y.
So, they are round, but they are green.
Round and green. That's a combination that didn't exist in the pure breeding grandparents. The grandparents were either round yellow or wrinkled green. We've made something entirely new, a round green pea.
Nature has remixed the album and the other 3.
It's the inverse. You get 3 boxes that are homozygous for little r, but have at least one big Y.
So they are wrinkled, but they are yellow.
Right. Another new combination. So we have 9 round yellow, 3 round green, 3 wrinkled yellow. That's 15 boxes. That leaves one single box left.
The lonely box in the bottom right-hand corner.
The one. And that's little r from the top, little r from the side, little y from the top, little y from the side.
But
Bars.
wrinkled in green. The full double recessive.
The one that looks like the recessive grandparent.
The only way you get this is if the pollen happen to be carrying the rare suitcase and the egg it fertilized also happened to be carrying the rare suitcase, and they found each other against the odds. It's a 1 in 16 chance.
So 9.3.3.1, this is the fingerprint. If you do an experiment and you get data that approximates this ratio, you know you're dealing with two traits that are sorting themselves out randomly.
Randomly is the most important word in that sentence, and that leads us directly to the big rule. Mendel's fourth postulate.
The law of independent assortment. It sounds like a spell from Harry Potter, assortio independum.
It's more powerful than any spell. It formally states that during gamete formation, the segregation of one pair of alleles is independent of the segregation of any other pair of alleles.
Which brings us right back to the suitcase analogy. The decision about the shape gene doesn't tell the color gene what to do. But I want to push you on this. Why?
Why?
Yeah. Why doesn't it? I mean, these genes aren't just abstract ideas. They are physical things inside the same cell nucleus. They're swimming around in the same genetic soup. Why don't they get stuck together? Why doesn't the big R allele say to the big Y allele, hey, stick with me, we're the dominant team?
That's a really good question. The textbook just says it's random, but it doesn't really explain the physical mechanism. The source notes mention meiosis, but it kind of glosses over the mechanics. It just says random alignment.
To understand this, we have to zoom way in. We have to look at the chromosomes. Remember, genes aren't just floating ideas. They are physical stretches of DNA, and they have a physical address. They live on a specific chromosome.
Okay, I'm visualizing the X-shaped chromosomes we always see in diagrams.
Right. So in meiosis, this is the first of two divisions to make sperm or eggs. Your homologous pairs of chromosomes line up in the middle of the cell.
Homologous pairs, that's the chromosome you got from mom and the chromosome you got from dad pairing up.
Right. The two copies of chromosome 1 find each other. The two chromosome 2s find each other. They all line up along the metaphase plate, right down the equator of the cell, ready to be pulled apart.
It's like a middle school dance. Boys on one side of the gym, girls on the other.
Actually, it's the opposite, and that's the key. Imagine the dance floor is the equator of the cell. You have the pair of chromosomes carrying the shape gene. Let's say mom's chromosome with the big R allele lines up on the left side of the dance floor, and dad's chromosome with the little r is on the right.
Okay. Big R left, little r right. Got it.
Now look at a different pair of chromosomes, the ones carrying the color gene. They're lining up next to the shape pair on the dance floor. Does mom's chromosome with the big Y have to line up on the left just because the other one did?
No. I guess it could flip the other way.
It's totally random. It has a 50-50 chance of lining up with big Y on the left or big Y on the right. The orientation of the shape pair has zero influence on the orientation of the color pair.
So when the cell finally splits down the middle to pull them apart?
The gamete that forms on the left side of the room might get big R and big Y. Or, if the color pair had flipped, it might get big R and little y. It depends entirely on how they all randomly faced each other on that cosmic dance floor.
That is independent assortment. It's not just a concept. It's a literal physical randomness in how the chromosomes stack themselves up before being divided.
Yes, but there is a massive catch, a huge exception to this rule. And the source material highlights this, sometimes just in the footnotes, but it's one of the most important concepts in genetics.
A catch. The rule isn't always true.
It's only true if the shape gene and the color gene are on different chromosomes.
Oh.
What if they're on the same chromosome?
So they are physically connected.
Imagine there are two houses on the same street. They're physically tied together on the same long strand of DNA.
Then they can't line up independently. They are handcuffed together. Where one goes, the other has to go.
Exactly. If the big R allele and the big Y allele are physically located on the same piece of DNA, then wherever that chromosome goes, they both go.
That completely breaks the suitcase analogy. You can't pick and choose anymore.
It's called gene linkage.
Yeah.
And if genes are linked, you do not get the 9.3.3.1 ratio. You get really weird ratios. You get mostly the parental types and very, very few of those new recombinant types.
So Mendel, was he just lucky?
He was either incredibly lucky or perhaps he was a brilliant editor of his own data. But the 7 traits he chose to publish on in P's were all on different chromosomes. Or they were so far apart on the same chromosome that they behaved as if they were independent.
He hit the genetic jackpot, because if he had picked 2 closely linked genes, the math wouldn't have worked out so cleanly and he might have given up and we wouldn't be having this conversation.
The history of science is filled with near misses and lucky breaks. But because his chosen genes were unlinked, we get that beautiful, predictable 9.3.3.1.
Okay, so we have the biology, we have the randomness of the chromosomes leading to this predictable 16-box ratio, but I have a confession to make.
What's that?
I hate the Punnett square. I truly, deeply hate it.
Class, you're not alone. I think every biology student on Earth hates the Punnett square.
It's so big, and the notes pose this terrifying hypothetical question. What if we want to track 3 traits? Seed shape, seed color, and, I don't know, flower color.
A trihybrid cross.
The math for that says each parent can make 8 different gametes, so the square would be an 8x8 grid. That's 64 boxes.
It gets worse. Don't stop there. What about 4 genes?
Don't tell me.
That's 2 to 4, 124 gametes, so a 16x16 grid. 256 boxes.
5 genes.
32 by 32. 1,024 boxes.
Okay, I am absolutely not drawing a 1,024 box grid. I barely have room on my notepad for this 16-box one. There has to be a better way. If nature is this complex, surely modern geneticists aren't sitting around with giant pieces of graph paper drawing squares the size of billboards.
No, they are not. And this is where we pivot. This is the moment where we stop drawing and start calculating, because it turns out you can ditch the square entirely if you just understand 2 incredibly simple rules of probability.
Probability. Oh no, it sounds like math class again.
It is. But I promise, it's the kind of math that makes you feel powerful. It makes you look like a wizard. It all comes down to the product rule and the sum rule.
Okay, sell me on this. How do I solve the dihybrid problem, the 9.3.3.1, without touching a pen to a grid?
Remember our cheat code concept, the idea of the 2 monohybrids.
That the traits are independent events.
Right. So let's just ask ourselves a simple question. We're crossing RR with RR. What is the probability of getting a round seed? Just the round phenotype. Ignore the color completely.
Well, in a simple 4-box square for just that trait, 3 of the boxes are dominant, 1 is recessive, so 3/4.
3/4. Perfect. Write that down. Now, a completely separate question. We're crossing Y with YY. What is the probability of getting a yellow seed?
It's the same logic. It's also 3/4.
Right. Now, here comes the magic. The product rule, which you can also call the A&D rule, says this. If you want to know the probability of 2 independent events happening together, you multiply their individual probabilities.
So if I want a seed that is round A&D yellow,
you take the probability of round, which is 34, and you multiply it by the probability of yellow, which is 34.
Okay, 3 times 3 is 9, and 4 times 4 is 16. That's 9/16.
Boom. There it is. The 9 from 9.3.3.1.
That took 5 seconds.
And you didn't have to draw a single line.
Okay, that is actually incredible. I feel like I've been doing this wrong my whole life. Let's try the rare one, the double recessive, the wrinkled green one.
Okay. What's the probability of getting wrinkled from an RRXR cross?
That's the 1 recessive box out of 4, so 14.
And the probability of getting green from a YXYY cross?
Also 14.
And we want wrinkled AD green, so we multiply them.
14 times 14 is 100 and something.
1 from 9.3.3.1. It works every single time. It works for 2 genes, 3 genes, 10 genes. You just break the problem down into tiny, easy pieces and multiply the results.
This changes everything. This means I can look at a trihybrid, that nightmare 64-box problem, and just do 3 little multiplication problems.
Exactly that.
Okay, so that's the A&D rule, multiply. But the source notes also talked about the sum rule, the OR rule. When would I use that?
The sum rule comes into play when you are looking for an outcome that can happen in multiple mutually exclusive ways.
Give me an example of what that means.
Okay. Let's say I ask you a different question. What is the probability of getting a plant with the dominant phenotype for shape, the round look?
Okay.
Well, there are 2 ways to get that phenotype, right? You could be homozygous dominant, which is big R, or AR. You can be heterozygous, big R, little r.
Right, both of those genotypes result in a round seed.
But you can't be both at the same time. A single plant is either one or the other. So because it's an OR situation, you add their individual probabilities together.
So the probability of being big R is 14, and the probability of being big R, little r is 24.
So you add them up, 14 plus 24 equals.
Which we already knew was the answer.
Exactly. It's just another way to get there. So the ultimate cheat sheet for genetic probability is AND means multiply, OR means add. If you can keep those 2 straight, you can solve ridiculously complex genetics problems in seconds.
One of the sources also suggested a visual way to do this that isn't a square. It was called the forked line or branch diagram method. It felt like a nice compromise between the giant square and doing pure math in your head.
Oh, I am a huge fan of the branch diagram. It's fantastic because it organizes the multiplication for you so you don't lose track of all the combinations.
Can you walk me through how to build one?
Absolutely. Visualize a list in your head. or on paper. In the first column, you list the phenotypic probabilities for trait 1. So you'd write 34 round, and then right below it, 14 wrinkled.
Okay, so I've got 2 items in my first column.
Now, from each of those items, you draw a little forked 2 branches sticking out to the right. On the top 2 branches, the ones coming from round, you list the probabilities for trait 2. So you'd write 34 yellow on the top branch and 14 green on the bottom branch.
And then I do the exact same thing for the wrinkled item below it.
You got it. You branch off wrinkled with its own little fork, writing 34 yellow and 14 green.
So now I'm looking at a diagram with 4 final endpoints.
Exactly. And to get the final probabilities, you just follow each path from start to finish and multiply as you go.
Okay, so path 1 is start at round 34, follow the branch to yellow 34, multiply them, 916 round yellow.
Perfect.
Path 2, start at round 34, follow the branch to green 14, multiply them, 316 round green, and so on.
It gives you the full 9.3.3.1 ratio in a nice, neat, organized list instead of a chaotic grid.
And I assume this scales up nicely? If you have a third trait, you just add more branches at the end of the second set?
It scales beautifully. It's the best way to handle a trihybrid or tetrahybrid cross without losing your mind.
We are definitely going to need that for the trihybrid section later. But before we get to the really hard stuff, I want to talk about the ultimate cheat codes.
Cheat codes.
Yes. Our source material provided some really specific, powerful formulas to calculate maximum possibilities instantly. No drawing, no branching, just plugging in one number. It's all based on the letter N.
Ah, yes. The power of N, where N stands for the number of heterozygous gene pairs in the parent.
Right, so let's test these out. For our dihybrid parent, RRYYN equals 2.
Correct. 2 heterozygous pairs.
Okay, Formula 1 from the notes is for calculating the number of unique types of gametes. The formula is simply $2.
This tells you how many unique sperm or egg combinations an individual can possibly make.
So for a dihybrid, ND2, that's 202 to 2, which is 4. That's exactly what we saw earlier. RY, 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 and RY. 4 unique gametes. It works.
Simple enough. What if we have a trihybrid, a parent that is AABCC, now N3?
So 2 and a 3 lars, which is 2 times 2 times 2, that's 8. It can make 8 different types of gametes.
And just to blow your mind for a second, a human has 23 pairs of chromosomes. If you were heterozygous for a gene on each one, N would be 23. You'd calculate $223.
That sounds very big.
It's over 8.3 million. That's how many genetically different gametes a single human can produce. It's a huge reason why you don't look exactly like your sibling, even with the same parents.
That is wild. Now, here is a trick question the source brings up to test us on this formula. What about the genotype AABBCC?
Ah, this is a good one. Be careful. What is N? How many heterozygous pairs are there?
Well, the AA pair is homozygous dominant, so that doesn't count. But BLIB is heterozygous, and CC is heterozygous. So N is still 2.
Exactly. So the number of gametes is 202 out of dollars, which is 4. Even though there are 3 genes involved, only 2 of them are actually creating variation in the gametes. Don't let an exam question trick you into using N3.
That is a very good catch. Okay, Formula 2. Number of phenotypes. The source says this is also $200 at least, assuming simple dominance like we have been.
Right, and a self-cross of a hybrid. So for our dihybrid, N2, it's $202, $200, there's 4, and that's what we saw. Round yellow, round green, wrinkled yellow, and wrinkled green. 4 distinct looks.
But then we get to the one that threw me for a loop. Formula 3. Number of genotypes. This is the code underneath the hood. The formula here is $3.
Yes, this is always a bigger number. There are always more genotypes hidden within the phenotypes.
But why 3? Where does the 3 come from?
Think about a single gene, like A. From an AXA cross, what are the possible genotypes the offspring can have?
You can be AA.
Brain possibilities. So for every heterozygous gene you add to the cross, you multiply the number of possible outcomes by 3.
So for a monohybrid, N1, it's just 3 other 1-1s, which is 3 genotypes. For our dihybrid, N2, it's 302 a 2, which is 9. Wait, there are 9 unique genotypes hidden inside those 16 bosses.
There are. You have RRY, RRYY, RRYY, RRY, RY, RRYY, and RY. If you count them up, there are exactly 9 distinct genetic combinations possible.
I never realized it was that predictable. It's like we've cracked the organism's source code.
That is the beauty of this math. It allows you to predict the true underlying complexity, not just what you see on the surface.
Speaking of prediction, the source material also mentions a way to reverse engineer a cross. Like you stumble upon a weird field of plants, you count the different types, and you want to figure out how many genes are controlling the traits you're seeing.
The detective work. This is a very practical skill for a working geneticist.
The rule of thumb, it says, is to look for the homozygous recessive offspring, the rarest type of all.
The one that is recessive for everything. The little r, a little r, a little y, a little y. The one in the bottom corner of the Punnett square.
Right. So the rule says if that rarest type makes up 14 of the total population, how many genes are involved?
Well, 14 points to a monohybrid cross. That's a 3.1 ratio. The one in that ratio is the 14, so one gene.
What if the rarest type is 116 of the population?
That's our dihybrid. That's the one in the 9.3.3.1 ratio. A 116 frequency for the double recessive tells you you're dealing with two independent genes.
And what if it's a really tiny fraction? Like you count thousands of plants and find that the double recessive is only 1 out of 64.
Then you are almost certainly looking at three genes. A trihybrid cross, because 14 times 14 times 14 is 164.
It's like carbon dating for genetics. You look at the rarity of the most recessive combination to tell you the complexity of the parents.
Precisely. It's a very elegant shortcut.
Well, you just mentioned the trihybrid. I think it's time we face our fears and go there. Section 6. Advanced applications and trihybrids. This feels like the final boss battle of the episode.
Let's dive into the deep end.
A trihybrid cross. Okay. We are adding a third trait to our P's. We have shape, RR, color, YY, and now let's add, say, seed coat color, gray versus white, with gray being dominant. Let's use GG.
Okay, so our F1 parent is now RRYYGH. 3 heterozygous pairs, ND3.
The full phenotypic ratio for this, if we were to write it all out.
Lay it on me. I'm ready.
Okay. We are crossing two parents with very messy genotypes. Parent 1 is ABBCCDD. And we cross it with parent 2, which is ABCCDD.
Okay, wow. That is a mess. We have 4 different genes, a mix of homozygous and heterozygous states in both parents. This is not a simple self-cross.
And the goal is to find the probability of getting a very specific offspring. Let's say we want an offspring that shows the dominant phenotype for all F4R traits.
So we're looking for a baby plant that has the phenotype of dominant A, dominant B, dominant C, and dominant D.
Exactly. If I tried to draw a Punnett square for this, I mean, I don't even know how big it would be. It would be huge, and I'd still be drawing it next week.
So don't even think about it. Break it down. Remember the rule. A tetrahybrid cross is just 4 monohybrid crosses happening at the same time. We tackle them one gene at a time.
Okay. Step 1, the A locus. We are crossing A from parent 1 with A named from parent 2.
Okay, A times AA. What's the chance of getting an offspring with the dominant A phenotype?
We know this one by heart now. It's the classic 3.1 ratio, so 34.
Good. Write down 34. Step 2, the B locus. We're crossing B from parent 1 with B from parent 2.
Ooh, okay, this is a test cross. Beeb times DB. If you do the little 4-box square, half the kids will be B, dominant phenotype, and half will be BB, recessive.
So the chance of getting the dominant B phenotype is?
1/2, 12.
Perfect. Write down 12. Now for step 3, the C locus.
We're crossing CC from parent 1 with CC from parent 2.
Well, parent 1 is CC. That means every single gamete it makes is guaranteed to have a big C allele in it.
Exactly. So it doesn't matter what parent 2 does. Every single offspring is going to inherit at least one dominant C allele.
So the probability of being dominant is 100% or just 1?
It's 1, correct. Write it down. Okay, final step. Step 4, the D locus. We're crossing DD from parent 1 with D from parent 2.
That's just the reverse of the B locus situation. A times a DD, again, half the kids will be DD, showing the dominant look.
So the probability is 12.
So now we have our list of 4 individual probabilities, 34, 12, 1, and 12.
And because these are all independent events happening on different chromosomes, we use the ND rule. We multiply them all together. Okay, 34 times 12 is 38, times 1 is still 38, times another 12 is 3/16.
3/16. There is your answer.
That took us about 45 seconds, and it was foolproof.
And you didn't have to draw a single box.
That is incredibly satisfying. It takes what looks like an impossible biology problem and turns it into a simple arithmetic problem.
That's the lesson. Huge, scary-looking problems are often just a bunch of tiny, easy problems stacked together.
I love that. Okay, we have conquered the math, but before we wrap up, I want to pivot back to the biology one last time, because Mendel was doing all of this with abstract factors and ratios. He didn't know what DNA was. He had no concept of a gene or a protein.
Not at all. He was working completely in the abstract. He saw the mathematical pattern, but he had no idea what the underlying physical mechanism was.
But modern science does. Section 7 in our outline covers what Mendel couldn't see. The source materials mentioned a fascinating 2010 study that actually went back and identified the specific genes that Mendel was unknowingly working with.
It took us about 150 years, but we finally found them.
Let's talk about his flower color trait. Purple versus white. The gene for this was identified, and they called it gene A.
Right. So Mendel just said purple is dominant to white.
Yeah.
But the modern question is, what is physically happening at the molecular level to make that true?
Researchers found that this gene A contains the instructions to build a protein called a transcription factor.
Let's unpack that term. A transcription factor is basically a master switch. It's a protein whose job is to fly around the cell and turn other genes on or off.
So gene A isn't making the purple pigment itself, it's the boss that tells the pigment factory to start working.
A perfect analogy. In the purple flowers, the pigment is a chemical called anthocyanin. The gene A protein works. It flips the switch on the anthocyanin factory genes, the pigment gets made, and the flower is purple.
So then what is wrong with the white flower? What's its genetic problem?
The white flower has a tiny mutation in its gene a single nucleotide change, a single-letter typo in its DNA code.
And that one little typo breaks the whole switch.
It's enough to change the shape of the protein, rendering it inactive. The switch is broken. It can't bind to the DNA and turn on the factory, so no anthocyanin gets made.
So white isn't a color paint being added. White is just nothing. It's the absence of purple because the factory is shut down.
Exactly, and that is a beautiful explanation for why it is recessive. If you have one working copy of the gene, the big A allele, and one broken copy, the little allele, the working copy can still produce enough functional switches to get the job done. You still get purple.
You only get the white phenotype if both of your copies of the gene are broken.
Precisely. That is the molecular reality of dominance and recessiveness. It's usually about function versus a loss of function.
And they prove this in what has to be one of the coolest experiments I've read about, the rescue experiment.
Oh, this is fantastic. They used a device called a gene gun.
Which sounds like something out of science fiction, but is apparently a real tool in molecular biology.
It is. It's a bit brute force, but it works. They take microscopic gold particles and coat them with normal, healthy, functional DNA from gene A.
And then they just blast it at the flower.
They literally blast it into the petals of the white flowers.
What happened?
The cells that got successfully hit by these DNA-coated gold particles, the cells that absorbed that healthy gene, suddenly started producing purple pigment. They formed little purple spots on the white petals.
They rescued the phenotype. They fixed the broken switch.
They did. And in doing so, they created this perfect, undeniable bridge between Mendel's abstract math, his 3.1 ratios, and the actual, tangible, physical reality of DNA, proteins, and molecular machinery.
That is a perfect way to bring the old and the new together.
It shows that Mendel was right, he just didn't know how right he was.
So we have traveled from a quiet monastery garden in the 1800s to a gene gun in a high-tech lab in 2010. Let's try to recap our journey.
We leveled up.
Yeah.
We moved from tracking one trait to tracking two, from the monohybrid to the dihybrid.
We established that iconic 9.3.3.1 ratio, the fingerprint of this kind of cross.
We learned that independent assortment means genes generally don't control each other's travel plans, and that it's all thanks to the random dance of chromosomes during meiosis.
And most satisfyingly, we threw away our giant grid drawings and replaced them with elegant, powerful probability math. Multiply for and, add for or.
And finally, we saw how Mendel's abstract factors were eventually revealed to be real, physical, molecular switches in our DNA.
Now, I want to leave our listeners with a final provocative thought. We spent this whole deep dive talking about how perfect the math is. 9.3.3.1. Independent assortment, clean ratios.
But I sense a but coming.
But the sources, and you dropped that hint about linkage. We built this entire beautiful mathematical palace on the assumption that all these traits sort independently. But you said earlier, if genes are on the same chromosome, these perfect ratios break.
They break down completely.
So what happens when the math fails?
That is where modern genetics really begins. Mendel, through luck or genius, studied the perfect cases where the math worked. But most of biology is messy. When the ratios are off, when you do a cross and you don't get 9.3.3.1, that isn't a mistake. That is a clue.
A clue to what?
It's a clue that tells you those genes are physically close to each other on the same chromosome. The more the ratio deviates from 9.3.3.1, the closer they are. It was by studying these broken ratios that scientists were able to map the first chromosomes. The failure of the simple math is actually the key that unlocked the human genome.
So the messy reality is actually more useful in the end than the perfect theory.
It usually is. The exceptions are always more interesting than the rule.
I love that. So the next time you look at a field of flowers, or even just look at the traits that run in your own family, try to see the equations, try to see the 116ths and the 316ths hiding in plain sight.
And be very, very glad you don't have to draw a 1024 box Punnett square to understand them.
Amen to that. Thanks for diving deep with us. We'll see you next time.